Good morning, everyone, and welcome to the Osage University Partners webinar series. My name is Bill Harrington. I'm the managing partner here at OUP, and I'm in charge of the life science investment activities. Today, we're going to address drug development and specifically the challenges of developing antibodies versus small molecules. Many of you have asked us, what do you need to do? What shouldn't you do as you put together packages around early stage efforts? And oftentimes, given the limited resources you have available, it's actually quite critical, what you should focus on and what you probably could get away without focusing on. So to do that today, we're going to have a conversation with Steve Holmes, who's the CEO of Capella Biosciences, which is an OUP portfolio company that has a license and an SRA with Sanford Burnham and is developing a number of antibody products. And Joe Vacca, a chemistry consultant for numerous startups developing small molecule therapeutics, including Cytier Therapeutics, a Jackson Labs spinout in which OUP is an investor. To get started, I'm going to ask my colleague Anurag Agarwal, who's really pulled this program together, to introduce our speakers and to take it from there. Anurag.
Right. Thank you, Bill. This is Anurag Agarwal. I'm an associate on the life science team and glad to be hosting this webinar. So as Bill mentioned, let me introduce quickly our panelists and then hand over to them to give their backgrounds. For targeting antibody and small molecule development, we have two experts in the field with decades of experience. First, we have Joe Vacca, who has a long history in small molecule development and has previously and currently is actually consulting for numerous startups like Quintus and Cytier, and previously was at WuXi AppTech and Merck, leading their global structural biology and CRO services at WuXi. For the antibody side, we have Steve Holmes, who's the CEO of Capella Biosciences and has been in antibody development space for a very long time. Previously, he was a senior director at Kymab and also a former CTO at Procure Therapeutics. Now I would hand it over to our panelists to quickly add a few words about them, and then we'll get started on our webinar.
Thanks, Anurag. This is Joe Vacca talking. So as you mentioned, I'm a medicinal chemist. I spent about 30 years at Merck Research Labs at the West Point, Pennsylvania site, which is just north of Philadelphia. And after I retired at Merck, I started working with WuXi AppTech, who is a company I'd worked with at Merck. And I was in charge of risk-sharing programs. And then I left WuXi about 3 years ago to become an independent consultant. And I've worked with a lot of small companies, helping them from the seed stage to get to their Series A stage by developing compounds and projects. So I'm glad to be here and look forward to the next hour.
Steve, could you maybe give a couple words?
Sure. Hi, this is Steve Holmes. So my background started really with in the pharmaceutical side with SmithKline Beecham and GSK, where I headed up all their antibody development and got a few antibody products into the clinic, one especially in the asthma field. I then joined a smallish biotech called Oxford Glycosystems, and again I was heading up the antibody engineering and antibody development side. That was later sold off to Celltech when I was there, and from there I joined another antibody company, this time developing fully human antibody fragments. That was at Demantis, which was sold back to GSK. And then following on from those, I've been in a Kymab, which is developing fully humanized mouse for making monoclonal antibodies. And for the last 3 years, I've been at Capella Biosciences, which I founded, developing therapeutic antibodies in oncology and autoimmune diseases.
Thanks very much, Steve. This webinar, we'll structure it as we always do as a panel discussion with Q&A, and we'll begin with high-level discussion around the development process, kind of compare and contrast challenges, followed by conversation and Q&A around specific areas where research and institutions can focus to extract maximal value from whatever asset they may be developing. As always, we really like questions and find that really animates the process. So if you'd like to submit a question anytime during the webinar, including right now, please use the Q&A feature on your toolbar. We will curate those and ask them at the time when it's most appropriate. And if we don't get to them, we will answer your questions, all of your questions, at some point after the conclusion of the webinar. You'll also see a poll appearing on your screen, and if you could fill it out, we always love to know who's attending today. So as we move on, therapeutics is really obviously primary focus for life science venture capital, and preclinical development of therapeutics is a very complicated, time-consuming, and capital-intensive undertaking. In the early stages of development, there are a lot of things that need to be optimized around various candidates to enhance drug-like properties. But given the limited time and resources that are available in an academic setting, it's critical to choose wisely and figure out what not only to focus on in the institution, but what will investors want you to have focused on as you present to outside venture groups or angel groups. So before diving into drug development and starting to spend money on what is a long and expensive journey, inventors really should ask themselves, what is truly feasible in a thinly funded academic setting? What's most valued by investors? And what should be done in-house or be outsourced? And overall, how much should it and could it cost? So before we dig into Q&A with the panelists, Anurag's going to present some data on what's going on today in small molecule and antibody development. There's a lot of text on the slides, and we'll be distributing them after the webinar, so we're going to move through them relatively quickly so we can get to our panelists. Anurag.
Thank you, Bill. Yeah, so here on the slide is depicted the overall FDA approvals for the last 20 years for small molecules and antibodies. And interestingly, if you notice, before 2000s, we had a steady decline in number of approvals. Following that, we had from 2000 to 2010, quite steady number of approvals, around 18 to 20. However, as everybody has noticed, there has been a rise or a raise in interest in therapeutics. And a part of it is driven by the increasing number of therapeutics approvals that we are seeing from FDA, with 2017 being the highest approval in the past decade. And the other key thing to notice here is that antibodies approvals have significantly risen in the last 5 years compared to a few in the previous decade to the more than around 10 for the last 5 years. And that usually seems to be the question. And so maybe, Steve, a question for you is there been any one big contributing factor that has led to this increase in the number of antibody approvals?
Yes, if I look back to the early 2000s, the ability to actually produce antibodies to scale and GMP manufacturing, the yields were very small. You need much less than a gram a liter. Whereas nowadays, I think the expression levels have gone up quite dramatically to about 5 to 10 grams per liter, and it makes the cost of goods much cheaper and the ability to dose in much higher doses. So manufacturing has been one big key factor in moving antibodies forward.
Thank you, and I think we will continue talking about this as we go through the development process. Before talking about the development process and the differences between them, I thought it would be good to review the overall difference between small molecule and antibody as therapeutic modalities. and this maybe most of the folks on the phone may already be aware of this, that small molecules are smaller, may have a limited specificity or variable specificity, can be delivered orally, and are usually cheap to make. On the other hand, antibodies are large biomolecules that are very specific, but are usually given by intravenous injections. And you know, while comparing these two modalities, they both have their positives and negatives. Having an oral delivery is desired, versus a long half-life with specificity of an antibody is also desired in a drug. So maybe the question that one can ask is, if given a very interesting target, how would you decide? Would you choose a small molecule approach or an antibody approach? And Joe, maybe you can take it first and say, you know, what makes you believe in a small molecule as the modality for a target over an antibody?
Yeah, I think your second bullet point there, as far as target, has a lot to do with whether you're going to look for a small molecule or an antibody. So if you have intracellular targets, most of the time you're looking for small molecules that can get into the cell. Extracellular, it could either be an enzyme or you know, a surface receptor, things like that. Certainly small molecules work and work well, and there's many examples on the market. And antibodies are also very extremely useful for this. I think you know, antibodies tend to have longer half-lives and don't have to be taken on a daily basis like a small molecule might have to be, and that's certainly many advantages, I think, go to the antibody approach. But I also think there's a bit of a specialty in making antibodies, whereas small molecule chemistry and safety has been around for, you know, 40 or 50 years or even longer. So it has a good track record.
Great. And maybe Joe, Steve, any additional points to add to that?
No, I think that's been quite well covered. I think one of the key things is the very unique specificity of antibodies. And I think, you know, they're definitely not intracellular targets. That is really extremely difficult to target antibodies to specific cellular processes inside a cell. So, you know, everything is dependent on the target.
So today, for the meat of the webinar will be focused on the development process and what the investor package should look like. So within the process overview, we will talk about what does this development process include, what are the activities, what are the key considerations when conducting these activities, and what are the challenges that are normally faced, and how much would it cost, and how long does it usually take to get from a target to an IND. In the investor package side, we'll talk about what a good investor package looks like. You know, how can academics include internal funding to address some of the, or progress their assets, and more also we'll talk about what should they not spend the money on given the challenges in those steps. And finally, we'll also discuss some external resources that one can use to push their assets internally, as numerous universities are considering these days, given the interest in therapeutics. And finally, we'll end up with some Q&A for everyone. So now to get started, you know, we would as Bill mentioned, the therapeutic space is a long, expensive process, and the process could be really divided into 4 steps, from hit identification to lead identification to dev candidate selection, and to finally to clinical candidate selection before the clinical trials can begin. Between small molecules and antibodies, these can take anywhere from 3 to 5 years and cost $7 to $10 million. Specifically, the $7 to $10 million is just the project cost and not any additional cost that a company has to incur. And again, on the antibody side, being a slightly more expensive endeavor than on the small molecule side that we'll get into further as we go into each steps. For today's webinar and for the time constraints, we'll focus on the first three steps from hit identification to development candidate selection, given that the clinical candidate selection is a more commoditized and standardized experiments that are required for an IND filing. So I'll just take a pause and see if any panelists have any comments on what, in this process, what they find most taxing and the most, the critical part of the development.
Well, from my standpoint, this is Joe. Before hit identification is certainly choosing targets and target identification and developing the assays so that then you can go into the hit identification. That companies struggle with that because there's a lot of attrition in targets at the early stage. So to me, that's the hard part. But once you find the target, then I think the next steps are developing the hit. So figuring out how you get your hit, you go to the literature and look for hits that are in the literature against the target if the target is known. Or if it's a new target, you have to somehow come up with a screen. And I know we're going to go into that at later stages. So I think the hit identification and lead to me, are a big part of the project because they will really affect the later stages of the project and development.
Okay. Thanks, Joe. Thanks for that. So as we dive into our process, the first step is the hit identification. And here, the two key activities that are to be conducted is to develop an assay through which can be used for a high throughput screening to find initial compounds, and also identifying a library that would be used to screen. And these two activities are consistent across small molecules and antibodies. So maybe we can start with antibody and Steve, what are the key things that you contemplate on before setting on a screening endeavor? And what are the key challenges have you faced in your career?
Steve, this is Bill. You were a little bit soft coming across last time. If you could speak up a bit, I think it'd be helpful.
Okay, sorry. Let's see if this is a bit better. Yeah, some of the key considerations. Well, first of all, it's which platform to use. And I think probably generating hybridomas is the most easiest and also the cheapest option to use. And the other one would be probably phage display libraries, so a library approach, but developing antibodies, and they could be fully humanized. But generally, that costs more. And in my experience, you may not be saving any time by comparing to hybridomas. The other key consideration is obviously antibodies are very specific, and normally depending on your target, you will probably only get a human-specific antibody. So really key considerations up front is what is my in vivo model going to be, and if it could be maybe something like a humanized mouse, or you might need to develop a human target knock-in. All these things must be thought of in advance before starting your project.
Right. And how often do you find that you have to develop a knock-in mouse model, because that seems quite an extensive endeavor from just the genetics perspective?
That is rare, I would say, because one of the key thing as well is that obviously if you're going the human target has to cross-react with the mouse receptors, so the odds of that happening could be quite remote. But in general, you could probably use humanized mouse models, and especially with the oncology situation, that's becoming much more common to use now.
Okay, great.
Joe, how have you seen screening libraries evolve over the last decade or so? I mean, there are a lot of options now, and trying to choose from among options and to work with CROs, or do you work with CROs? How do you access the screening libraries for small molecules?
Yeah, that's a good question. So, it used to be only big companies had libraries, of hundreds of thousands to now millions of compounds. Plus, they have the internal resources and robots to screen that many compounds. They have all the tools to call the hits and try to find the right lead compounds. But in the last 10 years, with the advent of development of CROs, they all have their own screening libraries, usually in the 150 to 400,000 compound range. Most of those libraries come from non-proprietary materials, compounds. So they're libraries that they either bought from companies like Cambridge and Enamine, and there are many companies now that sell libraries of 100,000 compounds or focus libraries against kinases, nuclear hormone receptors, etc. So you can either buy those and do things internally if you have the equipment, or you can go to a CRO and contract them to do the screening. Now, there's a cost there to do that. They usually take your assay and they'll develop it so that it can be done on, a couple 100,000 compounds. But it may cost you $1 to $2 a well per compound. So it could run up to the $500,000 range. And if you want to do DNA encoded libraries, there are companies out there that do that. And that's a sort of a specialty type company. And those are also pretty expensive to do those. I know there are a lot of academic institutions that now have their own screening groups internally. And, that's really great. You can take advantage of that. The other thing I thought of, there's a lot of companies now that actually have opened up their sample collection or parts of their sample collection to do screening collaborations with companies. So you could apply to have your assay screened using their screening library. It might be a couple 100,000 compounds. And I don't know what the reach-through rights are. It's sort of the deal that you do with the company is the deal that you do. But that's also that opportunity that may be a cheaper way to get your assay screened at a really good sample collection. Because usually company sample collections don't contain compounds that are out in the public.
Great. So, obviously finding the library is critical. What else have you, what are the key considerations you think of when you're thinking of developing an assay or, setting up the assay for a high throughput screen?
Well, I think you have to have a very robust assay that's reproducible, that is probably a quick turnaround. You don't want to have something that takes a week per cell, because you're going to be going through a lot of compounds, which takes a long time. So the other thing is, the hard part is, if you're developing a screen, a lot of times you want some sort of standard compound you can put in there, and if it's a new target, it's hard to figure out what the standard compound is. So you may have to buy a small library. You have their FDA approved compounds, for instance. A couple companies sell those libraries. They're about 2,000 or 3,000 compounds in the library. There's also some libraries. I once used one from Maybridge, which has 14,000 compounds, and it's covers a range of GPCR actives, kinases, etc. And 14,000 compounds is something you can do internally with a good pipetter. So I think that's the key thing. Once you get a compound or some sort of hit, a lot of times you could make your assay a lot better. You can improve your assay and then sort of go to the next iteration. Maybe that's what you use at the large company. The other thing to consider is, if you're talking to either companies or CROs, you ask them, for some advice, or hire a consultant that knows how to do screening to help out with the assays development. I think that that makes a big difference too. If you have people who have done this before in industry, that they can help your assay be a lot better.
Steve, one of the observations we've had over the years is with antibodies, there seems to be a little bit more, I'll call it black magic or art involved than maybe with small molecules. Maybe that's changing as experience grows. But do you have any perspectives on that and kind of where people tend to go wrong if they don't appreciate that?
Yes, there has been. You find a lot of CROs, for example, will always have their favorite method of generating antibodies, whether and the species they're using, whether you say you might get much higher affinity antibodies if you immunize rabbits. In my experience of all of these, it usually isn't the case. But one of the key considerations is that, for example, if you're generating antibodies via hybridoma route, is the immunization strategy is a bit of a black box. But one of the key things is always to have your antigen in a native formulation. So, for example, if you were trying to generate antibodies to a cell surface receptor, as part of your immunization strategy, you should always include a cell line expressing your target. That way, you're guaranteed to get a higher number of positives towards the native structure. And also, the other key consideration probably is in your initial screening as well, that again, you want to use, for example, like a cell line in your screening that contains your expressing your target, and also at the very early stage, if you need a certain species cross-reactivity, include that in your early screening assays as well. And the other bit of black box is generating immunization strategies whereby if you're using a mouse to immunize, you can get a better proportion of antibodies that are directed towards the mouse target as well. But those, I think, are key criteria that you need a lot of expertise in and would probably need somebody who's had experience in that to develop those strategies.
So for someone at an academic institution, a researcher, or someone in tech transfer, where can they find that expertise? I mean, some have those relationships, but many don't. Where can they turn for that kind of experience to help guide their initial efforts?
That is a very good question. There's some CROs that are exceptionally good, and they will give you some more unbiased views on whether their platform will be good for generating an antibody specific to your target. Whether there's a sort of a list of good consultants to go to for expert advice, I think you need to network around to find those people, depending on where you're located.
Yeah, it's a non-trivial undertaking.
Yeah.
So moving on to lead identification, once you've had your hits, then obviously there's still a lot of work to be done. The SAR efforts in this regard, Joe, can you give us any thoughts kind of on how that is evolving and becoming maybe more efficient, more effective, given some techniques that have evolved over recent years?
Yeah, I think the ability to do a screen and then be able to call the results using chemoinformatics, I think that's a big thing. So you can cluster a lot of hits into various buckets and then take a look at the buckets. The CROs I work with, they actually do it for you. They will do a lot of the MedChem effort, and they will evaluate the hits and give you a report basically telling you these are the hits maybe you should follow up on. And that's because they keep track of the physical properties of the compound, what they look, if they look drug-like or not. There's a lot known in the literature now about the hydrogen bond donors, acceptors. There's various rules that you might want to follow to use as guidelines. So I think there's lots of ways to pick out better compounds. You should also listen to your chemistry colleagues that are in the department. You may have some MedChem colleagues that have done this before, and bring them in as some sort of internal consultant to look at the chemistry if you're just doing total biology, and let them tell you which ones look like druggable molecules and which ones look like trouble. There's a lot in the literature now, a lot of papers written as far as what sort of compounds are good compounds and what sort might be what's called a PAINS, which is a pan-assay interference inhibitor. So there's a lot of different ways to call the results and try to figure out which are the best ones to then push forward to the next part of your funnel.
And one of the things we see surprisingly often with university-based efforts is, the declaration of a lead and sometimes even development candidates, in our opinion, is prematurely. There's been some work done, but it hasn't been particularly rigorous or robust. And, do you have a sense for what the minimum requirement is there? I mean, these are resource-constrained environments. In a large pharmaceutical company, you have the luxury of, I wouldn't call it unconstrained, but considerably greater resources to do this. Is there a happy medium or a hurdle that institutions should target as they go through this process, where they can say, okay, this is sufficient that we can move to the next step?
I think you've got a few things on the slide. So one is to, if you have clusters of compounds, you can look through and see, is there a structure-activity relationship that shows up, or are all the compounds one activity and, nothing gets better, nothing gets worse? So that would be a flat SAR, and you probably don't want to work with it. And then there are other compounds that you actually show some things that help and some things that hurt. And I would focus more on those type of compounds. Now, you're just talking hits at that point, and then you're in what would be called the hit to lead stage. And to me, a lead is when you start to get your compounds in the sub-micromolar activity range. Most compounds that become development candidates, if they're kinase inhibitors, they're usually nanomolar, same inhibitors, 5 to 10 nanomolar at most against the enzyme, maybe more potent against the cell-based assay, but still in the 100 to 200 nanomolar range. And they also would, by that time, they would have shown some good drug metabolism properties, such as metabolic stability. So if you have something that's in the 5 to 10 micromolar range in a cell-based assay, to me, that's still considered very early at the hit to lead stage, and it's really not a development candidate. You'd have to get something way down in potency, and as a development candidate, you have to have a lot of data on pharmacokinetics of your compound. So I think that's some of the challenges of doing academic research, is you don't have that medicinal chemistry support to bring in a couple chemists to do a lot of chemistry around your compound. Most compounds that come out of a screen are not a development candidate. I can't think of a single instance of where I've seen that in all the years I've done this. So it takes a lot of work to go from your hit to the development candidate.
Yeah, and we've seen the same. It's sort of an irreducible requirement that, hope isn't really the solution to. Steve, on the antibody side, a similar question, given a resource-constrained environment and some of the key considerations and challenges on the slide here, any advice for the tech transfer folks or the university researchers in terms of what they should try to focus on, given they can't do it all? And, what is kind of a minimal accomplishment that you can take forward and present to investors in this regard?
Well, in some respect, you're a lot luckier with antibodies in that the predicted PK/PD is usually very good with a long half-life, which could be a lot more different with small molecules depending on what you're trying to do with them. But the key really is to generate enough leads very early on to different epitopes. And you can do a lot of biophysical screening, which will help in your manufacturing process. But generally at a very early stage for the academics, I would say the key is to get a good large number of hits that satisfy all your key bioassays, and really to try and develop a human primary cell based bioassay as one of your very early screens. It gives you a lot of confidence in your antibody hits. And the other key consideration is usually if it's from hybridoma work, normally the affinity of your lead antibodies is adequate to start with. It's only when you start to use library approaches that you will be considered probably to do affinity maturation. So in general, hybridomas have a lot of advantages at very early stages. It's very easy to get the leads, and it's usually quite easy to get a good number of hits to feel very confident in moving forward.
And Joe, actually, we just got one question from the Steve. Sorry, we just got one question from the group asking if you had the option of progressing an IgG or IgM antibodies. Is there a preference of one over the other?
Absolutely. IgG, IgM antibodies are extremely difficult to manufacture at scale, and you have stability issues as well. So in general, an investor would definitely not favor an IgM product moving forward into the clinic at all.
Yeah,
fact,
and we can second that.
Absolutely. All right, just moving on to the next step, which is what?
Back. Okay. I just wanted to go back once for a second. In terms of challenges around this in vivo model, we talked about that earlier, Steve. You know, sometimes it's really challenging if you don't have species cross-reactivity. Are there any proxies or any ideas you have on what you can do if that's the case?
That can be a situation, yes. Usually, you may need to have some very good human primary cell assays to believe in your leads. And secondly, you might have to follow up with some primate work on doing PK/PD studies if you have exquisite specificity only to your human target, but the minimum you must have is a cross-reactivity to a monkey species, and that's usually cynomolgus. And that you usually have your cynomolgus target in your upfront screen as well, because that will be your tox species down the line.
Great, thanks. That's important to keep front and center.
Actually, Bill, that's another, brings up another question about your even small molecules, whether your compounds work against the mouse variant versus the human variant. So a lot of times investors, when they start to look at your project, they'll ask, well, what's the, does the mouse look like the human? Have you tested it against the mouse variant? Because they may show some in vivo efficacy in mice, but then find out that human, it has tenfold less activity. So that's something to always keep in the back of your mind, that you should probably look at the target in both species.
Absolutely.
Great point.
Absolutely.
Excellent.
Okay. Moving on to the next step in the process, the development candidate selection, where as Joe mentioned earlier, a lot of the work is done in improving the molecule, getting its target potency and selectivity, getting the right drug-like properties, and establishing good PK and tox profile for these compounds. Again, these activities seem very similar across both of the, both antibodies and small molecules. So maybe, the bigger question is as we are trying to, get to the potencies of like 1 to 10 nanomolar in biophysical assays, what are the biggest challenges that come across your table in trying to improve that potency? Joe, would you be able to comment on that?
Yeah. Yeah, a lot of the problems with trying to improve potency is either you don't have enough compounds being made on a weekly basis. So that means, you know, if you have one chemist, it's really hard to get more than 4 or 5 compounds a month, for instance. And then the other issue is if you have an assay that's run once a month, it's hard to, you know, stop what that chemist is doing once you find data out. So I think it's important that as soon as the compounds come in, you could actually test the compounds right away. That's an issue I've had with some projects and some small companies and academics I work with. There's not fast turnaround of data. And, you know, the other issue is that if you start with a lead that just shows flat SAR, you have to at some point make a decision to give it up and to move on to something else. So how do you find the right lead? And sometimes that means doing a little bit of DMPK work up front. You know, your basic assay might be do a microsome stability assay of some of your lead compounds, because some of them may fall apart pretty quickly, and some may be pretty stable or stable enough that you have a good idea that they may end up having good pharmacokinetics if you don't go too far away from the initial lead structure. And again, you could go to CROs and get the assays done at you know, a couple $100 per compound per species, and that sort of spend goes a long way, I think, in the future as you're working in the right series.
Great. Steve, you mentioned that, again, IgM antibodies are quite challenging to produce at scale, and that is something that is a key consideration of having at least medium-scale manufacturing process ready in this step. You know, could you further expand on, you know, what are potential ways to address those, or what are the most common challenges that we come across?
Yeah, one advantages of the going down the antibody route is a lot of the processes are commoditized now, and you can buy off-the-shelf kits. For example, a lot of work is done to assess manufacturing by just doing transient transfections, and there's some very good kits available, for example, from Invitrogen or Fisher Scientific, to evaluate your leads at a very early stage. And usually that is, are they, you know, medium to good expressors, or are they terrible? And you can just throw out the bad ones to start with. And there's quite a lot of movement towards testing some of the biophysical properties of antibodies at an earlier stage, and that is relatively easy to do with your leads in terms of looking at aggregation or stability. And the other areas which you can address at a very early stage is antibody engineering. And again, this is a really relatively straightforward now, with the number of kits available to academics to, for example, engineer the FC regions to get maybe a desired half-life, or if you want effector functions in your molecules. So those considerations can be done at a later stage, and they're very easy to engineer in, and also engineer different variants like various IgG isotypes if you want an effector function or you don't. Whereas again, a lot of kits available to do this, to evaluate your leads. So it just depends on what you want from your antibody and how it's reacting with your target. So in general, you know, it's oncology, you may want to have an effector FC, whereas in an autoimmune disease target, you probably want to have FC inactive. And that's just being very generalized.
Right. I could just, you know, kind of going further on that, you know, as antibodies are you know, delivered intravenously, you know, once a week or greater than once a week dosing is quite critical. How often do you run into issues that the leads do not have the right PK profile, and how difficult it has been to improve that?
I would say probably in the vast majority of cases, the PK of an antibody is related to its target. and it's not related to the antibody at all. So if you unfortunately may have a cell surface target that is internalized rapidly, and it's not an oncology indication, that could be that's very difficult to overcome that situation. You may have to do more frequent dosing. Obviously, in oncology, that might be an advantage if you wanted to put a toxic drug, for example, on an antibody. So it doesn't really quite target dependent. Unlike small molecules, antibodies are very predictable in their PK profile, and any issues are always target related.
Great. I think with that background in process, then let's move on to how we incorporate what we've just talked about in terms of what's presented, what the minimal acceptable investor package looks like, what do investors want to see, and what's reasonably achievable given the resources available in an academic environment. We talked about screening assays, having a lead compound, and then sort of the in vivo models and the challenges there. Joe, do you want to talk a little bit about what this package might look like from a small molecule perspective? What you need to have?
Yeah, I think you need to show that your compound, binding affinity 10 to 50 nanomolar sounds good, and if you have activity in a cell-based assay with some form of showing that you have target engagement. A lot of cancer compounds, you can look in cancer cells and then look to see that the right proteins are being affected. So I think that goes a long way with investors. The other part that you have down there is in vivo animal models. I think most investors always, if it's a new target, want to see some sort of animal model. It's almost always a mouse model, especially if it's a cancer project. And they'd like to see the compound work in that model. Now, they may not be so worried about the delivery method if you do IP dosing or sub-Q dosing. I think most academics don't have experience doing oral dosing in animals, so they'll do IP dosing and show an effect. What would be good is if you can somehow get plasma samples at the end of the study or in between and show that you are getting compound on board and you're, have coverage of your compound. That goes a long way. And that's also sort of a way to look for some sort of toxicity. If you're dosing animals for 3 weeks at a high dose and you're not seeing any toxicity, that's a good sign for your compound. And I think a lot of times people, at least in when I was at at places like Merck, they'll look for knockout models. So do your compounds still work in a knockout, whether or not you see toxicity, because that would show up as toxicity then a lot of times. So that might be another thing, is if you have the ability to have a knockout model to also dose the animals with your compound.
Great. And Steve, any similar thoughts on the antibody side? Like, what are the critical pieces of information you really have to approach investors?
There is another, I think, a key that is not really mentioned here, and it's probably exactly the same for small molecules. I think there is a big movement towards linking your target to human genetics, whether this is in sort of mutations or SNP analysis. I think that is vital, and that really helps in the downstream of making your target a lot more attractive to investors or maybe pharmaceutical companies. But as was previously mentioned for the small molecules, it's very similar for antibodies, really. Just understanding the potential toxicity of your target rather than the antibody molecule itself. But in general, all the in vivo models or in vitro assays are very similar in terms of how you'd get a good investor package between antibodies and small molecules.
Great. And then moving to the next slide, given that resources are tight, where should, and this leverages the discussion we just had, where should academics focus their efforts internally? Joe, I guess, again, we'll come back to you with small molecules. There's a lot of text here, but if you could kind of just touch on the high points. We have about 10 minutes left, but it's important, I think, to go through where to focus funding efforts and then also where not to spend money if you don't have much of it.
Right. So I think what you have listed here is a pretty good initial list. If you have enough money, you can try to hire 1 or 2 chemists. And I listed India or China. Those are the 2 main areas outside the U.S. where you would go. U.S. chemists would cost a lot more. So a lot of times you go externally. The issue is you may have trouble with your funding institution. Sometimes you have a limit that you can only spend the money inside the U.S. So that's one issue you might run into. In vitro ADME, again, you can do that externally, or there's a lot of companies in the U.S. that don't cost much more than going outside. And if you could put together a package and keep an eye on your compounds with some of the key pieces, maybe do a couple pharmacokinetic experiments in mouse or rats, and also look for hERG inhibition, so that's for QTC prolongation in the heart, microsome stability, that looks at metabolism possibilities in the liver, CYP inhibition, and near the end, if you have a compound that's pretty close to, not a development candidate, but pretty far along, you might want to do some off-target panels, and you can do those either in the U.S. or externally. Now, the in vivo models, some academic groups, I think, have good in vivo resources that you can run yourself internally. You can also go externally and have it run by people that do it on a regular basis. And they don't cost a lot of money. You just need to scale up compound. Sometimes it only takes a couple grams to do a mouse in vivo model. So I think it's worth going externally for some of those models. It's a more controlled setting than a lot of academic groups have.
Great. And on the antibody side, Steve, a few things that really are worth spending the time and money on up front?
In general, hybrid generation of antibodies is very straightforward. It's very cost effective. Okay, downstream, you're going to need to humanize those antibodies, but there's a lot of companies that are offering that work with number of different CROs now. You could go up front and screen with a fully human display library, but that is usually very expensive. And if you're not sure, or if you're limited in your funding and you want to get a lead as quickly as possible, then probably the hybridoma route might be the easiest way forward in terms of being very cost effective, and you're always got a very good way of generating some good leads.
Great. And then let's just flip then in the few minutes we have left to what maybe isn't the best use of funds, in this environment. Obviously extensive formulation work seems like an obvious one, but Joe, anything else that people might think is important, but, viewed through the lens of an investor or a potential pharma partner isn't so critical in these early days?
So I think a couple things. One is, like you said, running in vivo tox studies. So unless your compound is the development candidate or close to it, you're going to take the compound. An investor may take it and bring it inside a company, or you start a new company with it, which means you're just going to put a lot more money into it and look around for better molecules. So any toxicity issues you may have up front will probably be different in your final compound. So I really wouldn't do much on the tox side of things. Same thing probably for improving solubility or bioavailability. Unless you have a compound that disappears quickly, you probably don't have a good lead mop or a good development candidate for sure. But someone may look at your compounds and say, well, you've got a lead, it works in your assay, it's something I'm looking for against a new target, we'll take and improve it. And that's why people look at these things and they'll take them inside a company. It may take a couple more years to improve it. There was one, I saw a question online about is patentability important to people and to investors. I think it is important, but a lot of times you don't bring your compounds to the point where they could be patentable, or maybe there's a weak patent position around it. And again, companies will bring those in and probably develop other compounds around that. So it'd be nice to have good patentable compounds. I don't think use patents are really that useful to people and give them a lot of, comfort. So I think it's more compound matter patents. If you think you have a good group of compounds, that makes them more important to investors.
Great, excellent. And Steve, regarding antibodies, anything that folks tend to spend money on that may be better spent elsewhere?
I think the list that's up there on the screen at the moment is, I think, quite adequate. And there was one other question I think that's come through on how important is it to have epitope mapping before approaching investors. Yes, I think the more functional antibodies that you have in your package of leads, the better it is, and that also helps in the downstream as well. Obviously, the more leads you have, the better chance that you're going to get a very good expressing antibody in the development stage. So it's a numbers game, really. The better, higher the number of hits that you have that that fit your bioassay criteria, the better it would be, and also that would fit in with the investors feeling a lot more comfortable with your approach.
Excellent. Great. So we're just about out of time. Before we wrap, we'd certainly like to get everyone's feedback on today's webinar. We think we try to do a very good job, the best job we can, but we can always do better. So whatever comments you have, if you could fill out the feedback survey, we would appreciate it, and we'll incorporate your thoughts into future webinars. Joe and Steve, any final comments you'd like to make in the couple minutes we have left?
Well, I just like to comment. When I was with WuXi, I worked with a lot of small companies and academics, and you can do a lot with not a ton of money as you show up here, and you can find some people, I think, to help subsidize your work either through risk sharing projects. So ask around. And the other important thing is, I think, to go network around to people and talk to people. And that's one place you can find consultants, for instance, if you need to bring someone in to help you out. So, but I think we're in a good series, a good time right now with a lot of investor enthusiasm out there. Steve?
Yeah, I think Joe's mentioned some of the important points there. Is you've got to network around, definitely, to get some good advice. The more people you can ask, the better. And I think that's one of the key considerations.
Great. Well, Steve, Joe, thank you very much for taking the time out of what I know are very busy schedules to be our panelists for today's discussion. If any of our audience have any additional questions for our speakers or for us here at OUP, you can reach out to any of us directly. Emails are on the slide, which you should have a copy of. And thank you all for participating, and enjoy the rest of your day.